for use. Okay. Ultrasounds used for modulation of tumor progression and other addresses of cancer processes. We work in the group of ultrasonic resonators of the Institute of Physical Technologies of the Spanish National Research Council. As you know, ultrasounds in medicine are usually used for diagnosis based on two types of techniques. Ultrasonic imaging, which is based on the difference of acoustic impedances of different tissues or organs, which provides some reflection at the interface between these tissues. So it's widely used in every discipline of the medicine. And the ultrasonic elastography to obtain better resolution of some diseases or damage in a tissue, mainly related to cancer processes or other tumor tissues. But in therapy, we use also ultrasounds as a high-intensity focus ultrasound to perform ablation or other effects on the tissues which must be removed or replaced. But ultrasonic standing waves in microfluidics can be used for double application in cancer processes for early detection and actuation. This is named acoustofluidics. This technique presents some advantages as it's non-invasive technology, highly efficient because it works with small volumes of samples, and it means low-cost production as their manufacture is easy, simple, and not expensive. And as a result, they can be used for liquid biopsy. A combination of microfluidics and acoustophoresis gives rise to some applications that can be or are of interest for cancer early diagnosis processes. A combination of laminar flows with parabolic profiles of the velocity together with oscillating acoustic waves or oscillating Priser amplitudes established inside the channel of treatment provides a certain radiation force which drives the particles or cells toward certain acoustic equilibrium positions. This radiation force is produced by a nonlinear interaction between the incident wave and a wave scattered by the particle or the cell. And then following this expression, which relates densities, compressibilities, and size of the cell or particle to be driven, gives rise to different trajectories according to the approach of the particle cell to the position of acoustic equilibrium. It requires a time depending on different characteristics associated to the acoustic wave, as well as the properties of this cell. And of course, of the distance from the point of location of acoustic equilibrium to the location of the cell, the initial location. Then we have developed a Thinus chip. Now it's known as Thinus chip. It's a chip made up of a polymeric material, SU8, which is biocompatible, and it has two inlets and two outlets. In such a way that we infuse or inject the samples containing different cell populations to flow from inlet 2 to flow beside the channel walls. But through the inlet 1, which is just in the middle, we inject PBS or another fluid free of cells, which is biocompatible. Then, as we apply the acoustic field from a piezoelectric transducer attached to the chip, as you can see in in the photograph in the left side, we establish acoustic standing acoustic waves with a preserved node established just along the middle of the channel. So more susceptible cells, which in our case can be rare cells or tumor cells, epithelial tumor cells with longer, bigger sizes and less rigidity than the white blood cells, are more susceptible to be driven toward the preserved node located along the center of the channel. So they are extracted separately from their host suspension, which leaves the device through outlet 4 in laterals, the two laterals outlet 4. In these two images, you can see how first the small particles flowing just beside the walls before the acoustic actuation changed, and then collect along the center of the channel to flow just along it once applied the ultrasonic wave. Then we made the experiments with PANC1 tumor cells spiked in blood samples in which the red blood cells were chemically lysed, and then keeping the white blood cells. And then we made the experiments at some different flow rates, and then we measure the efficiency of capture or release of the tumor cells through the central outlet, and how the purity of this release was achieved at the different flow rates. So taking into account what white blood cells were delivered also through central outlet. And then we found that in any of the three cases, we found or we achieved a cell recovery of over close to 80%. And in the case of the flow rate of 80 microliter per hour, we obtain efficiencies higher than 80%, together with a purity of the same level, approximately. We found, we were analyzing the viability of the cells extracted from the device, and we found that the efficiency, as observed before, were varied with the flow rates, and we found an optimal flow rate in this type of chip, in this chip in particular, and the frequency and voltage applied to the ultrasonic transducer to achieve optimal results of cell separation. But now we wonder, later we wonder if low intensity ultrasound could be used for both early diagnosis of therapy and therapy. Ultrasounds traveling in tissues show or can generate thermal effects, experience attenuation, and depending of the near or far field, produce different effects. Parameters involved are frequency, dose time, and wave shape and continuity, pulse or continuous waves. The parameters involved for our experiments were limited to a frequency close to 1 megahertz. We applied times of irradiation of half an hour, 1 hour, and 2 hours. And continuously sinusoidal waves were applied with intensity close to 100 milliwatts per square centimeter. And in respect with the thermal effects, we have to take care of the heating effect. For it, we must take into account relation of temperature and its progression, not only in time or over time, but also depending on the intensity of the incident wave, attenuation coefficient of the tissue through which it's traveling, and the specific head of this tissue or material. Of course, depending on the incident temperature, initial temperature. So we use explants, pancreas tumor explants of mice, four mice. And we applied the low intensity continuous ultrasound in a cylindrical chamber resonating at 1 megahertz. And the low intensity ultrasounds were exposed for 4 different times, 30 minutes, 60 and 100 minutes respectively. And then my colleague Alba will explain to you our results.
Okay, thank you. So, to study the inflammatory profile, culture media was collected from controlled PANC1 tumor explants without ultrasound irradiation and from explants exposed to 30 minutes, 16 minutes, and 120 minutes of ultrasound irradiation. Then the main cytokines in inflammation response and immune system regulation were measured by using an ELISA kit. The results of the array show that after liquid stimulation, all the cytokines were regulated compared to control, non-liquid treated samples. Furthermore, in most cases, this upregulation is proportional to the time of exposure to acoustic irradiation. However, we can highlight that in the cases of interferon gamma, interleukin-4 and 5, it is not this way, since with 30 minutes of treatment, we obtain a higher upregulation than with 1 hour of irradiation. So we demonstrated that application of LICU on mouse pancreatic tumor explants results in significant upregulation of the inflammatory cytokines. As we have seen, LICU's irradiation for 2 hours induces upregulation of approximately more than 3 times in most of pro-inflammatory cytokines such as interferon gamma, TNF-alpha, and interleukin-1 beta and interleukin-2, while other interleukins with anti-inflammatory effects, for example, interleukin-4 and 10, had a lower upregulation. This suggests a pro-inflammatory response due to treatment with acoustic irradiation, although the possible anti-tumorigenic properties remain to be further explored. In addition, as mentioned, most cytokines show continuous upregulation with the ultrasounds irradiation time, and it would be interesting to study the optimal time of irradiation required to obtain the greatest beneficial effects. So to investigate the effects of LICU on the vasculature, as well as the collagen production of PANC1 tumor explants, the ultrasound-treated and controlled PANC1 tumor explants were fixed and labeled with CD31 and collagen-1, and then fluorescently labeled with secondary antibodies, green for vessels visualization and red for the collagen deposition. Cell nuclei were also stained with DAPI nuclear stain, and the samples were visualized by confocal microscopy. So no relevant effects were observed on tumor vasculature or collagen deposition and different times of application. So this result indicates that liquid treatment seems to be minimally invasive to the tissue structure and morphology. Okay, so otherwise, we have carried out monolayer experiments through cell culture of an epithelial carcinoma cell line derived from human pancreas, which is named PANC1 cell line. The main objective was to study cell migration under untreated conditions compared to samples with liquid irradiation, since it is known that collective cell migration plays a fundamental role in pathological processes such as cancer invasion and metastasis. During collective migration, cells move as a group, maintaining injection between cells, except for the some leader cells. Thus, migratory cell monolayers can be seen as tissues under tension. And it has been recently described that collective cell behaviors on monolayers can be altered by the application of external forces such as gravity or electromagnetic fields. However, the effect generated by oscillatory mechanical forces applied on cancer cells monolayer have not been yet analyzed in collective cell migration processes. Okay, yeah, the other one, thank you. And this is why we decided to perform bound healing assays in cancer cell monolayers. These assays were carried out by subjecting cells to different conditions. PANC1 cell lines were cultured in RPMI medium supplemented with 10% of fetal bovine serum for non-starving conditions where proliferation can be also observed. And in some other cultures, the medium was supplemented with 1% of fetal bovine serum to have cells in starving condition and only focus on migration processes. When cell culture reaches approximately 90% of confluence, the scratches were made by using pipette tips with a diameter of 2 millimeters. So the width of the bounds is about this size, depending on the pressure exerted. After making the bound, cells were supplemented with TGF-beta stimulation to promote cell movement in these in vitro conditions. So then PAN1 cell cultures were exposed to 10 or 20 minutes of Likus irradiation or maintain it in control condition without treatment irradiation. After that, the bound were cultured and photographed at regular intervals every 10 minutes for long periods of 24 or 48 hours. And we determined the bound coverage of total area and the average and standard deviation of the scratch width with the aid of ImageG plugin. Results of assays were obtained from at least 3 independent experiments. As we can observe, under control condition without Likus, the bound takes approximately 1 day to close, while both sonication doses keep the bound open during at least 48 hours after the sample exposure, being able to appreciate a greater effect at a longer time of treatment with Likus. Cells do not undergo any individual or collective movement during sonication or during the following hours, but they show altered dynamics several hours later during 2 days after the treatment. So we can confirm that long-term effects of Likus were observed on cell migration in PAN1 monolayers. And here we have an example movie of a bound healing process with and without ultrasonic irradiation. And this inhibitory effect induced of the cell migration was repeatedly observed in experiments. So in conclusion, the different studies carried out support that leaves can also modulate the inflammatory response of tumors, since we demonstrate that application of Liku on mouse pancreatic tumors plant results in significant upregulation of the inflammatory cytokines, and this could have further protection for antitumorigenic effects. And leaves can also produce a collective dynamic response of PAN1 tumor cells monolayer with an inhibitory effect on cell migration and probably proliferation. So these results offer a non-invasive strategy based on low-intensity ultrasound that open a door for a potential therapeutic tool for tumor progression and invasion. So thank you, and if you have any question or suggestions.
Yes, thank you, Dr. Gonzalez. Any questions from audience?
It's Mike Heller. I had one question about the wound healing work. Would this have any application when they do resections in pancreatic cancer? When they do the surgery, they'll do resections. So would this wound healing be something that might help in that process?
Excuse me, we don't understand your question. You mean that? Sorry? Could you please repeat us the question? We don't understand what you asked. Oh, yeah?
No, I wanted to know if the wound healing, you talked about wound healing, and I was asking if that process could be valuable when they do pancreatic cancer resections in surgery.
Yeah. Yes, no, bueno, please answer you.
With the sample of the...
Sí, directly. You mean instead of a cell kill too, but yes, is, bueno.
Yes, well,
this is the first step.
Yeah, this is the first step, but we at the moment we are performing only the in vitro condition, but as we seen an in vivo condition in these plants, the results. So the next steps would be to could analyze.
But even if it would be possible to take the cells from explant or resection, no, is what I understand that you ask. And then if the cells can be cultured, we could repeat this type of experiments, basic experimental studies with real patient samples processed in this way. Is what I don't know if we answered your question or not so much. But anyway, in the next year, we start with some in vivo experiments directly applying on alive mice which have induced pancreatic tumor, subdermal tumors. And then we will try to find, we have started in fact, but we will perform the study to know a real effect on in vivo samples, because the vasculature and other parameters and conditions which are not in the in vitro experiments can be affecting maybe favorably, but maybe not, who knows, so we don't know, the effects of the low intensity ultrasounds, which in principle are not harmful for the alive beings. I don't know if we have answered your question.
Yes, yeah.
Okay.